responses. Now, the way I want you to do this is type in your response and then look at the public chat pod and see how your response compares with your other classmates. Don't look at the public chat pod first. Type in your response. If you give the wrong answer, it's not a problem. Don't worry. If you give the wrong answer, no problem. We learn by our mistakes. But you have to participate. You're asking me, what's the best way to study? And the best way is to participate. You can't sit back and think you're going to take in all this information and spit it back out on the USMLEs. No, you have to participate. You have to be active in responding. And if only a few people respond, that does you no good. You have to respond. And if you give the wrong answer, we learn by our mistakes. We learn by our mistakes. My wife tells me that all the time. We learn by our mistakes. She's always correcting me. Just make sure she's not here. We learn by our mistakes. So please type in responses. Now, if a lot of you may have looked at videos and things like that. And if you have this stuff in front of you, if I ask you what's in, look at the videos and you have the answer in front of you, don't respond, because I'm looking for people who can recall things from other disciplines, things along that line. So again, participate, please participate, and you will see, trust me, that you will start to understand. Things will start to make sense if you participate. Here. Okay, so that with all that stuff out of the way now, okay, all that preliminary stuff out of the way, what we're going to do is, Eamon, please go slow. I can't go. I got only 11 days. Remember, this is a review. It's not the first time you've had this stuff. I'm trying to go through at a certain pace so I don't bore students. At the same time, I can't go too fast or too slow. I just have to go at a steady pace. Because this is a review, not the first time you've had this stuff. So I'll tell you, I'll do the best I can, but you have to keep it, you have to pay attention. I'm telling you right now, if you go away because you got a text message or you're going to answer something and you come back, you're going to be lost. You got to be paying attention. Don't worry about other distractions. Pay attention, okay? And you're going to do well. All right. Now, with that in mind, we're ready to roll. So what we're going to do is we're going to get into molecular biology for about 2 to 3 days. And in chapter 1 is a somewhat boring chapter. And the reason is, in chapter 1 is nucleic acid structure and organization. In other words, in this chapter, we're going to talk about and review what is DNA, what is RNA, what is a nucleotide, that type of stuff. And so it starts off as a somewhat boring chapter, but please bear with me for this first hour. Things start to get a lot more interesting, but we must cover the basics. You'll never understand chromosome stuff unless you know what DNA is, and you'll never understand DNA unless you know what a nucleotide is, what a nucleoside is, what a base is. That's what this chapter is, giving the fundamentals of stuff coming down the road. Okay, so now with that in mind, the first figure I want to talk about is nomenclature. There's a lot of nomenclature in this figure, a lot of terms that we need to make sure you understand the term. And here are 3 terms you must understand. And that is the term of replication, transcription, and translation. Now, for some of you, this is boring stuff because you've had it many times before. You're probably yawning at me. But for some of you, this may be you haven't had it for a while. It's okay. As long as you understand what I'm saying here is you must understand these words. The word the replication means taking DNA and making more DNA. The term of transcription means taking DNA, making RNA. And the term of translation means taking RNA and making protein from that RNA. These 3 terms, you must master them. On board exams, if they say to you, in transcription, a certain drug was used to treat some microbe, you got to know what transcription means. You got to know what replication means. There are drugs coming down the road that affect all 3 of these terms. and you must know what the terms mean. Replication, transcription, and translation. Matter of fact, we have a whole chapter. Chapter 2 deals with replication. Chapter 3 deals with transcription. Chapter 4 deals with translation. Each chapter, a lot of pharmacology, a lot of pathology associated with each of the 3 terms. You must know these terms. The sooner, the better. Master the terms. Now, you just had microbiology with Dr. Mascatello, and she talked about a term there called reverse transcription by the enzyme reverse transcriptase, which takes RNA and making DNA. Now, that does happen in certain microbes, retroviruses, but reverse transcription also takes place in the human body normally. And I'll stress, because the National Board asks this from time to time, I'll stress where in the body you have reverse transcriptase. But the idea is most of the time it is transcription. It takes DNA and makes RNA from that. Now, so chapter 2, 3, and 4 deal with each of these terms. Chapter 5 is another term that the National Board has been hammering medical students on, and that's the term called gene expression. How do you find a gene in your 46 chromosomes and turn it on? This term, gene expression, integrates. Why is it so high yield for National Board exams? It integrates with every discipline. Once we get to chapter 5, you're going to see the integration of biochemistry with pharm, path, micro, physiology, endocrinology, embryology, all coming up with gene expression. Molecular stuff coming up with all the other disciplines. High yield chapter. Chapter 5, modern molecular biology. Okay, now that doesn't come until tomorrow sometime. But before we get to that, though, we're going to start right at the beginning and talk about what is DNA and how does it compare with RNA. In other words, compare these 2 terms of replication versus transcription. Now, in comparing these 2 terms, replication and transcription, one thing is again, replication means DNA synthesis. Transcription means RNA synthesis. Now, before we talk about the differences between DNA and RNA in terms of structures and things, one thing I want to mention is that the sizes of the nucleic acids differ. The lengths of DNA can be different from the length of RNA. Now, what I mean by this is the first of many, many questions I'm going to ask you over the next 11 sessions. The first of many. Get ready with a public chat pod. Get ready with the responses. If I were to say to you that the DNA was taken from one of your chromosomes, the protein was removed, the DNA was then laid out in a linear arrangement. Suppose that DNA was stretched out and was this length. So if I take one of your DNAs, one of your chromosomes, get rid of the proteins, take the DNA in that chromosome and stretched it out. Remember, you have linear DNA. Your DNA is linear in contrast to bacteria, which have circular DNA. Your chromosomal DNA are linear. So if I took the linear DNA and stretched it out, suppose it would look like this. All right, here comes the question. What would RNA look like compared to that DNA? Again, don't go looking in the public chat pod first. You're not fooling anybody. You're not fooling anybody. Type in your response and then look at the public chat pod. What would RNA look like? Shorter, but how much shorter? How much shorter? That's right, well done. It would look like a little speck compared to that DNA. So then you ask, well, so what? The so what is, this has to do with chapters 6 and 7 in molecular biology. The idea is that when you want to analyze DNA or manipulate DNA, for example, when you want to do genetic testing of a disease, genetic testing for person identification, genetic testing in forensics, how often, like if you watch NCIS on TV, all those crime scenes, and they're talking about somebody's DNA is being analyzed. That my wife watches all the time, all these programs like NCIS, Miami, Chicago, whatever. But the idea is when you want to analyze DNA, because DNA is too large. How large is DNA, the chromosomal DNA? It's staggeringly large. If I took the 23 chromosomes that you got from your mother, 23 chromosomes that you got from your mother, and took the DNA from all 23 chromosomes and lined them up lengthwise. So I take 23 chromosomes that you got from your mother, took the DNA and lined them up lengthwise. You know how long that DNA would be? It would be a staggering 1 meter in length. Think about that. 1 meter in length is the chromosome, is the DNA that you got from your mother. 1 meter. So each of the chromosomes has, the length of the DNA is very large. And so the idea is that when you want to analyze the DNA, you can't analyze it directly because it's too large. So what happens is the DNA must first be fragmented. You cut up the DNA first, and then the DNA fragments are analyzed. And in chapter 7, we're going to be talking about a very important, powerful technique called RFLP, restriction fragment length polymorphisms. So you can analyze DNA after the DNA is fragmented. The DNA fragments are then analyzed on some sort of electrophoresis, southern blots, all coming up a little bit later in chapter 7. Don't worry about it now, but right now just focus thinking to yourself, man, DNA is amazingly large. Whereas RNA and protein made from the RNA, they are sufficiently small and they are never cut up. Never, ever cut up. You never cut up RNA or protein. You just had immunology with Dr. Muscatello in her great lectures. And in immunology, you never hear about protein being cut up in Western blots. In Western blots, you analyze the protein directly because protein is sufficiently small, it's never cut up. And RNA and protein, they're small, they are never cut up, never fragmented. Whereas DNA is always fragmented first. When you want to analyze the DNA, to analyze some crime scene or to analyze some disease like sickle cell anemia, we're going to be doing that in chapter 7. It'll be a family with a sickle cell trait. How can you analyze the DNA to determine the offspring, whether they are homozygous for sickle cell, heterozygous, or normal for the beta globin? All coming up later on in chapter 7. As a preview of things coming down the road. Hopefully you understand that that's all high-yield stuff. Now, another item here in comparing replication and transcription, and that is the phases of the cell cycle differ. The phases of the cell cycle in which replication takes place can be different than when transcription takes place. So that's what's coming up next. Is the review of the cell cycle, the all-important cell cycle. You must understand the cell cycle. High-yield cycle is the cell cycle. Now, you may have talked about it in other disciplines, but the idea is here is G0. Now, in G0, this is G1, S phase, G2, and M phase. Now, cells in the body which are in G0 phase, these are cells that are resting, non-dividing cells. Let me see your answers here. Let me see your answer. What's an example? In case the National Board asks you, what's an example of a cell type in your body right now that's a cell that's in G0 phase? What's an example? That's right. Y'all should be getting this. Neurons are in G0 phase. Skeletal muscle cells, cardiac myocytes are in G0 phase. Liver cells are in G0 phase. In your body right now, you have approximately 10 trillion cells. Think about that. 10 trillion cells, give or take a couple cells here or there. 10 trillion cells. The vast majority of them as an adult are in G